Professor Colin Hill is Professor of Microbial Food Safety in the School of Microbiology at the University College Cork in Ireland. He's also a founding principal investigator within the APC microbiome in Ireland, and which is a large research centre devoted to the study of the role of the gut microbiota in health and disease. His main interests lie in the role of the microbiome in human health, and he's particularly interested in the effects of probiotics and bacteriophage as well. So today Colin is going to be talking to us about the relationship between probiotics and the microbiome, help us understand the interaction between probiotics and the microbiome and some of the potential mechanisms. So over to you, Colin. Your slides look perfect, so I will disappear and leave it to you.
Okay, thanks, Louise. Thank you for the invitation, and delighted to be here. And yeah, the topic I've been asked to and chosen to speak about is the relationship between probiotics and the microbiome. And I think this is a really interesting topic because people conflate these two things, and they think that probiotics must somehow act through the microbiome or must interact with the microbiome in order to have their probiotic effect. And I want to kind of dispel that notion, but also show that probiotics can actually act through the microbiome. So there'll be some overlap with some of the previous speakers, but maybe some people will be watching this talk in isolation. So I think I'll just stick with the talk as prepared. So I did this about an hour ago while Sarah was talking. And what I'm showing here is that both probiotics and microbiome are very, very hot topics in research. Probably the hottest topics in microbiology, but one of the hottest topics in biology research in general. And for probiotics, as of this morning, there have been almost 30,000 papers published on probiotics. So there's a vast literature out there using this term. Of those, about 6,500 also come up when you search for probiotics and humans, and nearly 800 of those in 2020. So a lot of work is going on in humans, in individuals looking for these health benefits. Microbiome and microbiota is even more popular. There have been over 100,000 papers published, 20,000 or more in 2020. When I look back in 2013, there were less than 100 papers published on the microbiome. So that's an enormous increase, 200-fold increase in the number of papers published every year in the microbiome field. And you can see the kind of areas in which these papers are published on this graphic. And probiotics and microbiomes share many features. So obviously both consist of living microbes. Probiotics are delivered to a body site, generally, of course, the gut, but they can be delivered to skin or vagina or even lungs, while the microbiome is already present at these different body sites. Probiotics, hopefully, are highly characterized. So we know the organisms, we usually know their genetic content, we know their metabolic functionality and capabilities. The microbiome is extremely complex. And while we have kind of a bird's eye view of the microbiome and it's getting deeper and deeper, there's still a lot about the microbiome that we simply don't understand. And of course, probiotics, we experience those as a short-term exposure, as an almost an acute delivery of probiotics to the body site, whereas the microbiome is in long-term residence. Many of our microbes may become or colonize us during birth even, and stay with us until the day we die, hopefully many, many decades later. So these are permanent partners in our human biology. And of course, probiotics by definition must provide a health benefit, and we firmly believe that the microbiome plays a significant role in human health, a positive role in human health. At least a well-balanced, diverse microbiome plays a positive role in human health. So they share many characteristics, and it's obvious that people are going to confuse these a little bit at times. Now, in ISAP, I'm on the board of ISAP, the International Scientific Association for Prebiotics and Probiotics, and we've published a series of consensus statements around these biotic terms. You see them listed here, for example, prebiotics, probiotics, synbiotics, postbiotics, which is the most recent one, which only came out a week or so ago. And we also produced a consensus paper on fermented foods, which of course harks back to what Sarah and others were speaking about as well. And it's important to note that some of these interventions, these dietary-based, very often dietary-based interventions, work directly on the host. So probiotics could, for example, can work directly on the host, as could postbiotics and symbiotics, and some of them may act on or through the microbiome. Prebiotics, as we just heard about from Gemma act essentially exclusively through the microbiome. They could act directly on pathogens to reduce the risk of disease or to even prevent a disease. And all of these then, this combination of actions through the host, through the microbiome, through pathogenic organisms, act on the host and drive, hopefully, in the direction of healthy. So from sick to healthy, obviously. The consensus paper on probiotics was published about 7 years ago now. And a lot of the people here you'll recognize, I'm sure, and have been mentioned in the previous talks. And the definition of a probiotic is a live microorganism that, when administered in adequate amounts, confers a health benefit on the host. And this has achieved a lot of acceptance. Now, this is only a minor variation on the original WHO definition. And so this has been in existence in fact for more than 20 years. But this single paper has been downloaded over 53,000 times and cited over 3,000 times. So I think people have converged on this definition as describing what a probiotic is. The microbiome, on the other hand, is a much larger collection of microbes. You should always try to remember that gut microbes in the human body can measure at a level of about 10 to the 14. Whereas for a probiotic, you may consume it at a level of, let's say, 10 to the 9 or even 10 to the 10. But this is a 5-log difference. So probiotics are at a huge disadvantage in comparison to the microbiome in terms of having an impact on the body. But there are nuances to that, and I'll explain that a little later on. So what I would say is the microbiome is essentially the stage on which probiotics and prebiotics and symbiotics and postbiotics act. So here's the microbiome at the center. This is, let's, for example, use the gut. This is the gut microbiome. And the host obviously interacts with its environment through this gut microbiome to a very large degree. Most of our nerve endings and a lot of our immune system is focused in the gut, and therefore the host is very focused on this microbiome. Diet, we've already heard this morning, significantly can influence the microbiome, and Kevin and Sarah and Gemma really detail that in real detail. Probiotics obviously arrive into this microbiome. So this microbiome provides the stage, if you like, or the setting where host foods, including prebiotics and probiotics, meet. So it's obviously important. Probiotics do not interact with the host in some kind of sterile environment or some one-to-one manner. There is an involvement, obviously, of the microbiome. And quite obviously, the microbiome can be a source of probiotics. And many of the probiotics, the commercial probiotics they use, originated from human microbiomes or other microbiomes, but certainly they came from some microbiome somewhere. And so they come from these environments as well. Something that really bothers me at times is a kind of an inference that if probiotics are to work, they have to affect the microbiome, or they have to actually become part of the microbiome. They have to colonize. So probiotics do not, by definition, have to impact on host microbiome composition and our functionality. They may do, but they do not have to. They certainly do not have to colonize the host. And they do not have to grow or demonstrate metabolic activity within the host. They do not even have to have a known mechanism of action. They just have to demonstrate efficacy in terms of delivering a health benefit. But this can be controversial. There are 2 papers, I've listed their DOIs down here at the bottom, published in the journal Cell, which is one of the top journals in science. And it was represented as human gut studies question probiotic health benefits. And what this little précis of it here shows, researchers showed that many people's digestive tracts prevent standard probiotics from successfully colonizing them. So it was starting with the a priori assumption that you have to colonize in order to be effective. And that was even emphasized by the senior author here. We wanted to determine whether probiotics, such as the ones you buy in the supermarket, do colonize the gastrointestinal tract like they're supposed to. Now, I would object to that very strongly. They're not supposed to colonize the GI tract. And as it says down here, surprisingly, I'm not surprised at all, many healthy volunteers were resistant in that the probiotics could not colonize their GI tracts. And this was turned into a questioning of probiotic health benefits. So I think we have to be very careful that probiotics act in the environment where the microbiome is located, but they do not have to colonize and they do not have to influence microbiome composition or functionality. And that comes to kind of efficacy versus mechanism. This is an area which is obviously very exciting in probiotic science, but can cause some confusion. Evidence of efficacy must be generated in the target host. So it must be done in a human. If you want to market or sell a probiotic to a human, you must demonstrate in humans, not in preclinical models, that the probiotic has a health benefit. And of course, the regulatory authorities like EFSA and FDA will insist on that. And confirmation of efficacy then is essential to claim that something is a probiotic. And it must be in the target condition at the correct dose and so on. But mechanisms are completely different. Mechanisms are the means by which a probiotic might elicit a health benefit. And these are almost always determined in model systems because, of course, we can't do a lot of experiments on humans. We can do interventions, but that's a bit different from using molecular gene knockouts and dissecting the host after an administration of a probiotic, for example. And mechanisms are really intellectually satisfying. And most scientists, and of course most industries working with probiotics, want to get to mechanism because mechanism can help you to select better strains, grow your strain better in order to deliver its effect. But it's not actually essential for developing a probiotic. But most mechanistic work is done in mice, rats, preclinical models. And sometimes people are a bit disparaging and say, oh, that was only done in mice, or that was only done in rats. But if it's done to elicit mechanism, I think that's entirely appropriate. If it's done to elicit efficacy, then it's not appropriate. And probiotic efficacy can have a number of different levels. Some probiotics may all have the same effect. So there may be a lot of effects. For example, things like gut barrier reinforcement, impacting short-chain fatty acid production, where a lot of different probiotics may share the same effect. There can always be a core benefit of ingesting live, safe microbes. Some probiotics may have multiple effects. They may affect things like bile salt metabolism, affecting cholesterol levels, for example. They may neutralize carcinogens. They may synthesize vitamins. They may have a number of different effects. And then some may be very strain specific. So there may be a probiotic that elicits an effect that essentially no other probiotic has been shown to do. And so again, it's important when we think about probiotics that we recognize there may be some common benefits, some frequent benefits, but then some very select benefits. So if you're taking a probiotic for a specific condition, you should be very careful to make sure you're taking one with the best clinical evidence in that condition. Here's an example of widespread efficacy. Here was a meta-analysis done by Ritchie and Romanek back in 2012. So this is quite old data, but they looked at 74 studies, 84 trials, over 10,000 patients with a range of different probiotics. And what they will show was that in general, probiotics are beneficial in treating and preventing GI diseases. And as it says here, across all diseases and probiotic species, positive significant effects were observed. All age groups, single versus multiple species, treatment lengths. So it was an overall positive effect of probiotics on kind of general gastrointestinal health. And this reinforces the suggestion, at least, that for a lot of probiotics, there may be these general health benefits. Here's another example for acute diarrhea. Again, a meta-analysis of a lot of different trials. And what they showed was that probiotics significantly reduced the risk of antibiotic-associated diarrhea by 52%. But here the effects were not maybe as widespread. The effect was dependent on the age of the host, so that's a host factor, but also the genera of strains used. What they showed was that certain genera had a better effect over other genera. So these are kind of widespread, but not core benefits of probiotics. And then you have some which are extremely strain specific. So here, the ESPGHAN working group for probiotics and prebiotics looked at the evidence for a range of different probiotics for preventing nosocomial diarrhea in children, and they found evidence that only one of the probiotics that they were able to get enough data for actually demonstrated a benefit. So there may be some that are strain specific. So if you're looking for a general GI benefit, maybe you could use virtually any probiotic. If you're looking for an acute diarrhea probiotic, it may be more limiting. And if you're looking for something in children, prevent nosocomial diarrhea, maybe there's only one probiotic with sufficient evidence to drive that choice. Now, the potential mechanisms by which probiotics can act have largely been determined again in animal models. Some very small number have been determined in humans. But of course, probiotics in this nexus again of microbiome, host, and diet driving health, probiotics can act through all three of these vertices, if you like. They can act through the diet. Probiotics can act on dietary components and generate something like short-chain fatty acids, which could have a beneficial effect on health. Probiotics can act directly on the host, through the host immune system or nervous system, contacting with the brain. We've heard a lot about the brain-gut axis. Or, of course, they could influence the microbiome. While prebiotics, antibiotics, fecal microbiota transplants, they act essentially through the microbiome, or at least primarily through the microbiome. So probiotics can have multiple mechanisms. And these can be a result of multiple mechanistic or effector molecules. So this is a paper we published a couple of years ago in Current Opinions Biotechnology, showing that some of these molecules, for example, probiotics can deliver things like bacteriocins, enzymes, metabolites, structures, just simple bacterial biomass. And some of these may already be delivered in the initial dose. So the probiotic, the dose that you take on consumption, may already have all of the active molecules you need to derive the health benefit. You may not need metabolic activity or growth or certainly colonization in the microbiome to elicit the effect. And some may require active metabolism in the GI tract if they're going to produce short-chain fatty acids, for example. Here's an example of host response. This really nice paper by Michiel Terbezem and his colleagues showed that when they looked at biopsies of human tissues, this is really going in and taking biopsies of people, volunteers who've taken 3 different probiotics at different times with washout in between. And what they were able to show when they looked at the immune response in these biopsies, that different strains elicit very different responses. So here's Acidophilus casei and Rhamnosus, and they elicit very different immune responses. Here's a kind of an immune panel, a cytokine readout. So obviously not all strains are the same, and they're not all perceived identically by the host immune system. And interestingly enough, some of these transcriptional responses actually overlap with response to various drugs. So for example, the Lactobacillus rhamnosus transcriptional response overlaps with the drug kephaline, which actually promotes bowel movement, and rhamnosus has been shown to have an impact on gastric transit. So we may even get a hint as to mechanism through these kind of studies. We're more interested in our group on probiotics and infection, and we again see multiple mechanisms. Probiotics may act by directly interfering with a pathogen, by directly killing it or downregulating virulence factors, for example. May act through immunomodulation, as we've just seen. The probiotic may interact with the immune system, making the environment more hostile to invasion by a pathogen, for example. Or they may act through the microbiome, increase the presence of safe organisms. They may temporarily colonize and block interaction. They may increase mucus production, tight junctions, so through barrier function or exclusion. And we have shown at least one example of this where it's direct antagonism. So here are 6 different probiotics. We're only showing one animal here, but obviously this was done in multiple animals. 6 different probiotics were fed to these 6 mice, and then the mice were infected with Listeria monocytogenes. And this Listeria monocytogenes was engineered to emit light, and we could follow the light through the body of the living animals. These animals are all alive. There's a little nose cone here by which they're being anesthetized so we can take pictures of them. But you can see that 5 of the 6 probiotics simply do not work. The Listeria is reaching the gut, it's growing, then it's infecting the liver and the kidneys, and then eventually it's a lethal infection for these animals. But one probiotic is completely protecting the animal from infection. So a life-death outcome, a true probiotic, albeit a murine probiotic. This has not been demonstrated in humans. And what we're able to show was that that particular probiotic, UCC118, produces a bacteriocin which can kill the Listeria. And this gave us an obvious potential mechanism by which the lactobacillus might be preventing Listeria infection. But to prove that, what we were able to do was we made a knockout of UCC118, a genetic knockout which no longer produces the bacteriocin. And when we tested that in the animal model, again, you see infection in the animals that got no probiotic, no infection in the animals that got probiotic, and infection restored in those animals that got the bacteriocin negative probiotic. So one of the very few instances that I'm aware of where a single molecule has been and a single mechanistic explanation for a probiotic effect. And probiotics can be turn out to be very, very important in preventing infection. This is a lovely paper published 4 years ago now in Nature by an Indian group led by Pinaki Panigrahi. And what they showed was that a synbiotic, a combination of Lactobacillus plantarum and fast fructooligosaccharide, very large trial, 4,500 infants in rural India, and they observed a 40% reduction in death and neonatal sepsis. An amazing outcome for a simple administration of a synbiotic, a probiotic and a prebiotic. So these are probably very important. I want to shift a little bit now because I want to talk about the relationship between probiotics and the microbiome. So I want to talk about your microbiome. And your microbiome is assembled in you from the moment of birth. It's assembled from all of the bacteria you came into contact with, and you selected out some of those bacteria which have now become essentially your permanent partners throughout life. So whether you were born by C-section or vaginally, whether you were fed breast or bottle, your genetic background and so on, they will all influence the assembly of your microbiome in early life. And then you have that microbiome essentially throughout life. It can be influenced, of course, by diet, antibiotics, disease state, things like that, but essentially it's pretty stable. And then as you age, your health deteriorates and your microbiome kind of collapses a little bit and loses that stability that we observe throughout the healthy part of your life. And of course, we know that your microbiome plays a role in health and disease. These are mainly associations, of course, to date, but we end up with about 1,000 to 2,000 species that are unique to each individual. And evidence has been provided for associations with a range of diseases. This is by no means a complete list, but a lot of people suffer these modern diseases. These diseases cause a lot of morbidity or mortality, and the microbiome is associated at least with many of these different disease states. And the microbiome is complex. Here's a paper that we published here in APC some years ago, again, showing that the complexity of the microbiome is very difficult to capture in 2D images. But what we were able to show here was that the healthy elderly living in the community had one type of microbiome structure, and as they moved into long-term care, that microbiome structure changed and diversity was reduced. Diet obviously became less diverse, and of course people became more frail and became more sick. These again are associations, but the evidence is growing that if we can reverse that deterioration in the microbiome, we can reverse some of these deteriorating health effects as well. And again, very complex, noisy symptoms. These are people, healthy controls. These are people with ulcerative colitis, and then people with Crohn's disease. You get shifts in microbiome structure, like we saw the shifts in the elderly. You get shifts in things like diversity and complexity and richness of the microbiome, but they're very noisy signals. It's not like probiotics. There generally don't seem to be single microbes just simply driving disease. It's composition and functionality of the microbiome. And I would also emphasize the point that you have many microbiomes. We have obviously skin, oral, vaginal, lung microbiomes. We have gut microbiomes, but even within the gut, we have many different microbiomes and different densities of bacteria throughout these gut microbiomes. So for example, here's a recent study we did, it's not published yet, in macaques, so another primate, and what we were able to show was that these are family levels. These bars, each bar represents a different family. Members of the kind of size of the bar represents the relative abundance of that family at every site. And what you can see is that as you go from tongue to stomach, through the small intestine, proximal, middle, and distal, small intestine, cecum, proximal, middle, and distal, colon, the microbiome changes constantly throughout that journey. So when we talk about the microbiome, generally we're talking about this. This is the fecal microbiome, the distal colonic microbiome. But a probiotic may be acting back up here in the middle portion of the small intestine. And so judging the impact of a probiotic on the microbiome, if you're looking down here, may not be a good reporter. And this is emphasized by this image here. Probiotics are tourists, they're not residents. So you can look at this in two ways. You could say this is an example of the upper GI tract and the lower GI tract. And this might be a probiotic coming in as a tourist in the upper GI tract, plenty of room to look around and engage with the environment. But in the GI, in the lower, in the colon, really, you can't even find the tourist anymore among these resident bacteria. So it's much harder to elicit an effect in the colon than in the upper GI. And again, just to emphasize that, if we look at the density of microbes throughout the microbiome from tongue through to distal colon, you see that the number of bacteria differs wildly throughout that journey, if you like. And probiotics, of course, dilute as they go down through the GI tract. And so again, in the upper GI tract, it may be that probiotics outnumber the microbiome and by log scales, but in the colon, the opposite is true. So again, we need to be a bit more precise when we talk about probiotics interacting with the microbiome. Which microbiome are we talking about? And to kind of emphasize, this is a study from our own group, again from quite a while ago now. What we did was we isolated 10,000 strains from the microbiomes of salmonella-free pigs in infected herds. These pigs were living among other pigs that were salmonella infected, but they were not infected. And we reasoned this could be due to their microbiome composition or structure. And so we isolated 10,000 strains. We screened them one by one for in vitro anti-salmonella activity, something like the study we did with the listeria in the mice. And eventually we identified 5 strains with in vitro efficacy. We call this the 5 Live Consortium. It was a TV program in Ireland called 5 Live at the time. And that's why we picked that kind of faintly ridiculous name. But there are 4 lactobacilli and a Pediococcus pentacetus. And one of those produces a bacteriocin, but it's not active against salmonella. And what we did then was we fed those that 5 strain mix to pigs. We first confirmed that these pigs were salmonella free. We gave them about 4 by 10 to the 10 mix of these probiotics for 30 days. And on day 6, 7, and 8, we challenged these pigs with Salmonella typhimurium PT12. Now, this is not an easy experiment to do. To challenge large animals with a pathogen that causes diarrhea is no fun for anybody involved in the experiment. But what we saw was that the control pigs, 80% developed diarrhea on day 12. So 4 days after the salmonella was administered, none of the pigs given the 5 Live developed diarrhea on day 12. Clinical scores, these were blinded and judged by vets. The clinical scores were significantly down in the probiotic fed animals. They were shedding several log-fold fewer salmonella, and they put on more weight than the control pigs, which of course is an important performance indicator for pig farmers. But interestingly, when we looked at these 5 strains in the pig feces, we found that one of them, the Lactobacillus murinus, completely dominated the fecal microbiome. And we might have assumed from that this was the most important of the 5 probiotics. But what we could do with pigs, which of course we could never do with humans, was the pigs were sacrificed when we looked in the other regions of the GI tract. And in the ileum, where of course is where salmonella actually infects, it was a different bacterium that was the most dominant one. In fact, it was the Lactobacillus salivarius that was the most important. So again, we would have made a significant mistake if we used the fecal microbiome as a reporter for the ileal microbiome. Now, ingested microbes, I've said they do not have to influence microbiome composition in order to have an effect, but of course they may influence microbiome composition. And here is a simple experiment where we used heat-killed lactobacilli, postbiotic if you will, because we didn't want the lactobacillus itself to grow and to influence the microbiome. We just want to see the impact of introducing this bacterium biomass, essentially. And what we saw was that on day 0, the 2 groups, these were in the murine models, these are mice, two groups of mice could not be separated in terms of their microbiome composition. And then over 8 weeks, the microbiomes significantly diverged. So these are the animals got the heat kill lactobacillus. These animals were control animals. And you can see that the influence of a single bacterial species directly influenced these, the microbiome within these animals. It also reduced colitis. These animals were actually a genetic breed which developed colitis, and it also had a positive impact on colitis. And of course, you can start to add these and say, well, it has an impact on microbiome composition, and it has an influence on colitis. 2 and 2 is 4. The impact must be via the microbiome. But of course, we have not demonstrated that. We have not proved that. These could be coincidental rather than causal. Another example here, taken from the literature, not from our work, Lactobacillus plantarum P8 was administered to 33 individuals for 4 weeks, again at about 10 to the 10 per day. And this resulted in an increase in specific short-chain fatty acids, in propionate in particular, across an acetate, across different age groups. No increase in butyrate. But here you see the influence of a probiotic on the functionality of the microbiome in eliciting the increased production of short-chain fatty acids. This was not linked to a health benefit, so formally this cannot be called a probiotic in this particular experiment, but it does confirm that probiotic-like species can influence microbiome functionality. And here's an example of Lactobacillus casei shirota, a strain familiar to many of you. This was fed to overweight children in Malaysia, and this led to significant alterations in gut microbiome composition of both Lactobacillus and Bifidobacteria species, and also significant differences among total short-chain fatty acids and propionic acid among overweight children. Again, this wasn't linked to a specific health benefit, so we can't call it a probiotic for this particular, for any health indication on the basis of this experiment, although of course it is a probiotic for other reasons. But it does show that ingested microbes can influence microbiome composition and functionality. Here's a very important study, I think, that I don't think it doesn't get enough attention. This is a paper published in Science, and it showed that the gut microbiome modulates response to anti-PD-1 immunotherapy in melanoma patients. These are the so-called checkpoint inhibitors, the real wonder drugs in many cancer treatments. But many people do not respond to these drugs. And what they will show was that if they transferred the microbiome from responders to mice, they also respond to the treatment. While if you transferred it to from non-responders to mice, they did not respond. So the microbiome essentially was responsible for whether you respond to microbiome or to checkpoint inhibitor treatment or not. Now, there's a second paper published back-to-back with that one, which showed the exact same thing. Microbiome composition can predict the 3-month response to these wonder drugs, but they also went on to show that responders had higher levels of Akkermansia mucinifila, and when they supplement Akkermansia mucinifila to non-responder feces, it restored the efficacy of the drug. So obviously Akkermansia mucinifila presents itself here as a significant candidate probiotic with a health benefit that could have enormous consequences. And it essentially acts through the microbiome, and comes from the microbiome. So double whammy there. And we did publish a paper a couple of years ago again on this whole topic of next generation probiotics. The existing probiotics are, of course, the species we're very familiar with, but I think the next generation of probiotics will be Akkermansia, Bacteroides, Faecalibacterium. These are already being developed, and they will possibly be used preferentially in medicine and maybe less so in food and supplements. But also I think we can expect that genetically modified versions of the standard probiotics, if I can call them that, will find their way into medical treatments as well. So I want to conclude by saying microbiome and probiotics, they are in a relationship. That probiotics can improve human health, there's no doubt about that. And the microbiome is the setting where host, food, prebiotics, and probiotics meet. And therefore it is obviously important. And I don't want to understate its importance. But probiotics in the upper reaches of the GI tract will completely dominate the microbiome. They will be tourists, they'll only be passing through, but they may outnumber the microbiome by several log factors. And therefore, they may play a very important role in just simply directly interacting with the host immune system, enteric nervous system, and even with the diet and those reaches of the gut. Some probiotics certainly will influence microbiome composition and functionality. Some mechanisms for some probiotics may well be via the microbiome, but that's not a prerequisite for microbiome efficacy. And I would obviously re-emphasize the microbiome is also a very valuable source of probiotics. And so it's very important for those reasons. And lastly, if you'll indulge me on this day, this paper was published only yesterday, only last night, so it's very fresh on my mind. I want to just pay tribute to Todd Kleinhammer. I think Todd is someone who had an enormous impact on probiotic science, and particularly in introducing an incredibly high degree of rigor to probiotic scientists, an outstanding experimental scientist, sadly missed. He passed away only a month or so ago. And so I'll finish by thanking Todd for his influence on the entire field and for his influence on me. And he beat me up regularly on the squash court, and I thank him for that as well. So I thank Todd, and I thank you for your attention. Thank you very much.
Thank you ever so much, Colin, for a lovely presentation. I think we have a couple of questions that Rafi will pose to you, and I have a few to ask as well. So I'll hand over to Rafi first.
Perfect. So the first question we have for you is, should there be guidance on probiotic intake for the elderly where their diet diversity reduces? We often promote eat anything to gain weight, improve appetite, reduce risk of malnutrition without thought of diversity in our gut microbiome.
Yes, I would agree 100%. I think I would temper it a little bit by saying I think it's very important that elderly people maintain a diverse diet, because the diverse diet will support a diverse microbiome. You will not you can only increase the diversity of the microbiome even if it colonizes with a probiotic by one species. So, you know, giving a single species to somebody with maybe many hundreds of species can only increase the diversity very marginally. But I do believe that probiotics can change the environment in such a way that it would promote diversity and retain and preserve the diversity of the microbiome. I would very much encourage elderly people to maintain a very diverse diet, but also to maintain a diet high in microbes. Fermented foods, not processed fermented foods, but fermented foods containing live microorganisms. And I think probiotics are a very easy way in the elderly to get a reasonably high dose of live microbes delivered in the diet. I think unfortunately, as elderly people grow old, obviously they lose sometimes the joy of food. And very often they move into long care residential homes where maybe the diet is a little bit mundane or routine. I think rather than bringing an elderly relative flowers or chocolates, bring them yogurt, bring them cheese, bring them something that will restore the microbiome, that will bring live microbes in the diet. And I think you'd be doing a lot better than chocolate or flowers.
Wonderful. And then, are there any key drivers causing the highly individual response to probiotics that is often seen in human intervention trials? Do we get responders and non-responders? Yes,
absolutely. And it's a critical issue. Remember, though, this is true also of all medicines. With virtually every medicine, you saw the example there with the checkpoint inhibitors. Some people respond, some people don't. There's a thing called the NNT, the number needed to treat for most medicines. And the NNT can be as high as 10, 15, even 20. Sometimes you need to treat 20 people with a drug for one person to benefit. And the same is true of probiotics. And the reasons are likely to be many. Genetic basis, the diet, the type of microbiome you already have, the nature of the probiotic, the health benefit you're looking for. So yeah, I think it's inevitable that this is going to be very complicated, and there will be responders and non-responders. We shouldn't regard that as a failure of probiotics. I think that's true of virtually every intervention that has ever been tested in humans.
And then the last question we have for you is, you talked about the importance of determining the health impact of probiotics in healthy individuals. What effects could we reasonably expect? And would it be sufficient to use interim markers such as blood sugars, appetite response to vaccines, rather than disease outcomes?
Yes, absolutely. I think, we have general indicators of health, cholesterol levels. There are biomarkers, cholesterol, blood pressure. There are many biomarkers of health. And so maintaining health during stressful times, for example, maintaining lower cortisol levels during a stressful event. There's a lot of things, interventions we can do in healthy humans that demonstrate a health benefit. A health benefit doesn't have to be clinical. It doesn't have to be something that's measured in a clinical trial. It can be measured in an intervention study or an observational study, and show that people could even be as literal as, quality of life. We saw that in some of the earlier presentations. Do you feel better? Do you feel, are you more comfortable almost? Are you more relaxed having had this intervention? So I think we have to see that health is a very broad canvas, and we should interpret it pretty liberally, I think.
And we actually just had one more question pop up. If our microbiome is so much greater than a probiotic, is multi-strain probiotics better than a single-strain probiotic in influencing the host microbiome and health?
No, I mean, a priori, again, you can't just say multi-strain is better. There may be, instances where two different probiotics in a mix act together, or there may even be examples where two probiotics in a mix act against each other's function. I'm not aware of any instance of that, but of course it's possible. I think, the bottom line every single time is, if you're taking a probiotic for a specific health condition, look for the one with the best evidence in that health condition. Maybe if you're just taking a probiotic as a general, you just want to ingest more live microbes, then a multi-species one may be the best one to choose.
Wonderful. I don't know, Louise, if you have any more questions.
I do have a couple. So you showed a really nice slide that showed how our gut microbiota varies depending on the location within the gastrointestinal tract. So my question is, do you think we'll ever understand where a probiotic strain is having its effect? Because we can't look at that in human intervention studies.
Yeah, that goes to the kind of efficacy versus mechanism argument. There have been studies, like the study that Michiel Clarebezin did, where they went in and literally took biopsies up along the intestine and looked at the transcription response in the intestine at the site where the probiotic is at its most, is most abundant. But these are very, you wouldn't do that just casually. You might do that if you're looking for a probiotic that is having an impact in cancer biology or some, very serious disease with a very serious morbidity or mortality outcome. I think it's going to be a real challenge for us. We can do it in animal models, we can get hints, but yeah, it's going to be an enormous challenge, because even within, we've seen that if we take two biopsies a centimeter apart in the GI tract, in the upper GI tract, we see quite different microbiomes, a centimeter apart. And so a probiotic acting across that kind of canvas, very difficult to know where it's actually eliciting its effect, unless it's through an effector molecule that we can, reproduce the health benefit by just introducing that molecule, for example.
Okay, lovely, thank you. And then I have one more question, just to link us all the way back to Sarah's talk at the very beginning about consumers' understanding. We see quite often negative articles in the media around probiotics. So there's some a couple of years ago saying probiotics are quite useless. So it makes quite a complicated setting for probiotics in the consumer environment. So I just wondered what your thoughts are on kind of the media's interpretation of the research and some of those negative articles.
Yeah, it's difficult, because we're trying at the same time to educate people about the microbiome and the importance of the bacteria, and that they play this important role. And then we've got probiotics kind of getting mixed up in that. I have a clinician colleague who always addresses an audience with the following question. He says, if I asked you, do pills work? What would your response be? And people go, well, of course pills work, but you'd have to have the right pill for the right condition. He said, well, that's my answer when people ask me, do probiotics work? And I think very simple examples like that, if you're going to use a probiotic, you should only expect it to work if you're taking the right one. You wouldn't take a pill for a headache to reduce your cholesterol levels. So you'd recognize immediately that was a very foolish thing to do. So I think the messaging has to be very simple. And, the probiotics are an acute intervention. The microbiome is your kind of long-term partner. And if we can get that clear in people's minds, I think we'll have gone a long way.